Thank you and welcome. Thank you so much for being so numerous this evening. I would like to introduce the two speakers. We have Professor Martin Antic, Director of the Synthetic Biology Laboratory at CBio, the Department of Cell Computation and Integrated Biology at the University of Trento. And then we have a guest coming from very far away, from Melbourne, Professor Alan Dorin, Associate Professor of Information Technology at the Monash University, and also belonging to the Monash, the Association of Information Technology. So let's start with you, Martin. So we just had this fine introduction. I know that you deal with artificial life and synthetic biology. We had a short talk earlier on. It's a very fascinating world, but also a very complex one. So in very easy words, which I can understand, what is synthetic biology and what is the role of synthetic biology in ACDC?
For the question, and thank you for, Lucilla, thank you for hosting us here this evening, and thank you all for coming to listen to this discussion. We will try to speak slowly so that you have the translation coming in real time. Yes, it is a complicated story to try to make it precise. Synthetic biology has two main meanings. One of the main meanings is to synthesize life. So to think about what life is in its fundamental components and then try to make it in a laboratory, in a biological laboratory, to kind of re-synthesize life. The other main meaning of synthetic biology is to take living organisms that life has produced, nature has produced, and engineer them in a new or different way for a specific purpose. For example, to produce food, to produce energy, something different than what they normally do. So there are two approaches in synthetic biology, two main approaches. The project ACDC, which comes from an Australian band, stands for Artificial Life with Distributed Cores. Artificial cells with distributed cores. So artificial cells is related to the first definition, how you build a cell artificially in a laboratory. Distributed cores is when you think about how life is organized into a hierarchy. So you have, a cell membrane, and inside the cell membrane you have specialized compartments. We try to make those types of things in the laboratory. So we try to control how you make a cell membrane and then make the specialized compartments that can do specialized functions. Taken from biology, so we try to make stuff that's similar to biology in order to make a completely synthetic cell. That's the project, ACDC.
Ci torniamo tra pochissimo e vediamo qual è.
So let's now see the connection with the work of Professor Dorin. So Alan, what about your field, which ranges or straddles halfway between artificial life and ecology, computer science and nature instead of synthetic life? Can you tell us what it is?
Thanks for the question. And thanks for the translation. So, as you pointed out, my area is in technology and computer science, but I'm primarily interested in the interface between technology and ecosystems. And so when I think about artificial life, I'm thinking now about how the things we make change the way living things interact with one another and with whatever it is that we've put out into the world. And one of the simplest examples of that might be something that you've come across in the media in the last few years, a robo-bee. Now, I'm against robo-bees, for the record, but there are about 10 to 12 labs around the world that are trying to build a micro vehicle, a very small unmanned automatic vehicle that is like a little drone that can potentially pollinate flowers. And there are at least a dozen reasons I can think of why that's a really stupid idea. But as an example of technology that changes the way ecosystems operate, I think that's a very good one, because it pollinates flowers, it does so in a way that's different to biological organisms, it competes against the biological organisms, and it selects flowers to pollinate in a way that's different to the way a natural biological insect might. So that's one way I think now of artificial life.
Facciamo un po' a test.
Okay, let's go back to Martin. Well, I seem to understand that your work is aimed at understanding how a living system works by recreating some specific characteristics. Why? What is the purpose of such a research project?
Asking for my opinion is we see that biology is a very powerful force, of course, on this planet, and it has come up with many interesting solutions to challenges. For example, that you find life everywhere. Everywhere you look on this planet that has liquid water, whether it's far under the ground or in the sky, you'll find living organisms. So life has found a very good solutions to all different types of challenges, environments, and we have much to learn from biology. So the motivation here is, okay, we have very limited understanding of the complexities of nature, but we have some understanding. So we say, okay, let's take some of our understanding, try to make some experiments to resynthesize some of the properties of living systems. So it is for sure motivated by curiosity, basic science. We're not trying to solve the world's problems with these experiments, but we need to start somewhere. And we think it's an interesting way of, let's say, taking inspiration from nature to come up with new technologies that could be useful, beneficial for the world. This is the reason.
Da un certo punto di vista.
So, from a certain viewpoint, we are very close to the notion of origin of life.
Because we think the approach to take is to start simple. Start simple and build some complexity. So in that way, it's a bit related to our concept of how the origin of life started. But I like what Alan just said about, in general, what we're working with are artificial things that humans create. So in the experiments of AC/DC, we don't have to say, well, you know, this is the way life maybe has started on this earth, which we don't know, and we only follow this path. No, we are happy to take any artificial way of reproducing life. This is no problem for us. It could be completely artificial, and like you said about the pollinators, they're still pollinating, but in a different way. So we can make maybe an artificial cell, but it's working in a different way. This is no problem for us.
E quindi, tornando.
So, going back to Alan, as there is this complexity and also some urgency in your field, so what is the aim, what is the purpose, what is the aim, what is the priority for you personally also?
So, like my friend here, I'm absolutely fascinated by living systems and have always been. And I add to that, as you said, an urgency. I see the opportunity to work with artificial living systems in the way I'm thinking about them, as explained before, to enhance the capability of the natural biological world to manage itself in the face of the mess that we're making. And so my goal is to try and find ways that technology can assist natural living systems to become sustainable and to become self-sustaining. And I think that's quite a different goal to many in my field, because my aim is to put technology in to learn something so that we can pull the technology out and let the living system manage again by itself. So rather than putting the technology in as a band-aid or a stopgap or something that must always be there in order to keep the planet ticking along, I want to learn about how the planet operates and try and fix the things that we've damaged, and make it so that we can all live, not just the humans, but all the other animals and plants and fungi and bacteria. Many of them don't have a problem, but some clearly do.
E prima
Well, earlier on, we were talking about an aspect of your approach that I like very much. We were discussing about the fact that, in your opinion, the actors to be involved in your field could also be farmers, for example, those who actually work on the field and in the field. And something that you don't like very much, I understood, is when an engineer or a scientist tends to forget about the work, the actual work of those who have to do with bees, for example, but it can be many other things. So why is it fundamental, in your opinion?
We need to feed the planet. People need food. The population's growing, the climate's changing, and as a result, industrial agriculture is growing, and yet a large proportion of people on the planet survive on farms that are less than 2 hectares. So something like 75% of the world's farms are less than 2 hectares. Pretty small farms when you consider in Australia we might have a single field that's 250 hectares of something like canola or wheat or something. And industrialised agriculture is destroying habitat and changing ecosystems, but surprisingly, really, ironically, it's dependent on the sustainability of the natural ecosystems around agricultural fields. And so as we industrialise our agriculture and as our urban developments continue to sprawl and grow, we're removing natural habitat. And so as a farmer, people are kind of caught in this seemingly, anyway, a conflict between the push to modernise what they're doing and the push to sustain and maintain the natural environment. So I see my role as finding ways that technology can be used sustainably to show how industrial agriculture, because I do feel we need industrial agriculture, how it can be managed more effectively. So I'll look at applying technology in a way that a farmer can learn better how to manage their natural and agricultural environments for food production that's more reliable, uses less chemicals and damages the environment, has a smaller environmental footprint.
Grazie. Quindi torniamo alla biologia sintetica.
So let's go back to synthetic biology. In order to have a general view, well, we have understood what you are doing in your research work. But basically, what is life from the viewpoint of a regenerated type of life? Does it have to do with DNA? And what does it mean to recreate it synthetically? So we have to understand this notion.
I think the fundamental question is what is needed to create this kind of artificial life that we're speaking about here in any sort of context. It seems to me one of the main components is this ability to evolve. This seems to be a really crucial part of what we're speaking about. And so we are far away from that as far as the technology. So in this point, we are recreating some of the things that we, that are less complicated. What that means is, can our systems produce necessary molecules so they can function? Yes. Can they produce energy so they can sustain themselves? Yes. Can they move around in their environments to find resources? Yes. These are all artificial things that we can create in the laboratory. Can they evolve on their own? No. They're not advanced like even like a bacterium is. They're very, very simplistic things. And so in that way, there are some advantages because you can use, right now we're just exploring with science, but you can use engineering principles as well. Say, okay, I have a specific application, I have a problem to solve, I need to create a specific molecule in these cancer cells so it affects the growth of the cancer cells. And then we can say, well, we can try to create something like this, and it's not alive, so it's not as advanced as the cancer cell itself. But it is, again, it's inspired by biology, it's producing molecules, it's producing energy, and hopefully it's producing some sort of active compounds at the right time, right place, to modify the growth of the cancer cells. This is the dream of this. So on one hand, the technology will be limited because it's not evolving. So that's the limitation. But then again, that's also maybe good because we're now producing a technology that is pretty well under control and has a specific purpose and doesn't do anything else besides that one purpose. So that's the reason.
Ok, la mia domanda era...
Actually, my question was a bit easier in that it seems to me that we are moving away from the definition of life that we took from our school books at my time. Now research has reached a point where the definition of what life was 20 to 30 years ago has changed.
I think there's a paper published where I think they have identified more than 200 different definitions of life. So we are...
Una complessità estrema.
Yeah.
So an extreme complexity.
Saying our idea about life is changing over time, but I think it's diversifying. It's diversifying. It's not becoming more crystallized into something very central. That's not necessarily a problem. The kind of work that we're doing is all done in a laboratory, which means that we can have a hypothesis and we can test it. We can say, for example, do we think that the ability to move from one place to another is important for life? Well, let's make the experiment where we have this going, and we could analyze that without 4.5 billion years of evolution behind it, which is what organisms will give us. So we could watch organisms move from point A to point B, but they have their own agendas, which are billions of years old. These technologies are new. We're making them in the laboratory, and then we could isolate specific properties and say, okay, what is interesting about this? What does this particular thing do? And we do not have the complexities of evolution, for example, to kind of muddy the waters. So to get back to that question, that means my hope is by doing these types of experiments, we can clarify better what are living systems and what are non-living systems. And even if that is a necessary distinction, I'm not sure. My personal view, I'm more open-minded about what this is, and I don't bother too much about the definition of life. Because I think we have, besides, I don't know what you think about this, but besides biology, we might have other versions of life that we've already created on this planet.
Can I jump in?
Of course,
So,
please.
even though it was a long time ago, my PhD thesis was partly on the definition of life. And at that time, I was convinced that there would be this absolute definition and that there may have been 100, 200, however many, one of them would be the right thing. Yeah. And I settled on this idea called autopoiesis, which came about in the 1970s. And it defines life as a chemical system that is responsible for creating and maintaining itself, its own own structure. Basically, it's a metabolism that builds an organism. It's a set of reactions. And at that time, I thought, this is the most important thing to understand, and how do these systems build themselves? But as I mentioned before, in the last 20-something years, I actually don't think that's the most important thing anymore. I don't mind if someone calls this life or this life. To me, what matters far more is how something we build interacts with our ecosystems. And so it doesn't matter to me if you're talking about a mobile phone that sounds like a bird when you hang it up in a tree and convinces birds to sing. It doesn't matter if it's a chemical system that imitates a bird or a robot bird. What matters to me is that the ecosystem's responses are changed in and around that piece of technology in a way that alters their usual operation or interactions. And for me, that's what life is about, I suppose. And so even though I might not define life as a mobile phone, if it's interacting with ecosystems in a way that changes the way they operate, then it doesn't matter to me if you call it life or not.
Molto interessante.
This is very interesting, and I realized that even though we are talking about science, we have been talking a lot about philosophy and technology. We have spoken about technology. So, given this direction in our conversation, I ask you a question. In relation to recent years and to the fact that both your work in the 2010, 5 years have accelerated enormously your field of work, also due to the development of technology, I guess, in practical terms, so that major steps forward have been made. So my question comes now, what is it that in recent years has accelerated your field of work so much, and how?
A field, I guess some people call it big data. So the idea is that technology has allowed us to analyze systems, big systems, collect lots of data on these systems, and then once that came, we needed the computer-aided tools to analyze what the big data is telling us. So it just seems like the pace of science has increased rapidly, as you say, because I think the technology has allowed us to gather more information and to do more analyses. But what is not necessarily also accelerating is our understanding of these systems, which is interesting. So we're collecting a lot of data, which is actually quite costly. And it seems like, well, what do you do with that? What kind of hypotheses do you have? What do you want to understand about the systems? And it is helping, but I think that the rate of data accumulation is very fast because of the technology, because we have the machines to do this. But our understanding of biological systems is still coming at a very, very slow pace. So that's quite interesting as well. So technology is a bit outpacing. So that's one thing. And then just from the synthetic biology side, it has now become easier to do genetic manipulations of existing organisms. So not just collecting data on natural systems, but taking natural systems and changing them, specifically with some things like CRISPR, which many of you have heard about. It's a genetic engineering tool. You change the genome, change the DNA with CRISPR, and you look at the outcome of these things. So we're actually able not just to collect data, but we could change something about the living system and understand what has happened. What did the change do to this living system? So we can start to change things a bit. I think both of those together have been very crucial in this field of synthetic biology.
Alan, e anche nel tuo caso?
Alan, what about your case? So is it so that there is a different speed also in your field?
Martin's talked about are important in the work that I do. I think if I had to kind of pinpoint one or maybe two things, though, that arguably would be more relevant to my work than, say, Martin's work, one would be computer vision. And maybe computer vision as an example of new sensor technology. So as soon as you're able to perceive the environment in new ways, collect new data, you're able to build devices that interact with that environment in a much more complex way than before. So if your system can't perceive a change, it can't respond to that change. So an example being computer vision and AI and deep learning technology enables us to identify what a camera sees to the species level in many cases. So an insect flies by and my team is saying that is this species of insect, for instance. Another example would be hyperspectral imaging. So it used to be that a hyperspectral camera would cost tens and tens of thousands of dollars. It's now possible to get smaller devices for thousands of dollars. Those enable you to tell exactly how much light is reflected from a surface at a particular narrow frequency. And that data is valuable for satellite imaging to the point where you can tell when a particular flower is opening. You can use it at a small scale. So we use a spectrophotometer that enables us to understand the spectral reflectance properties of a petal on a flower. So we can see exactly what light is being reflected from a flower petal just in the lab with a pen, basically. Now, why is that important? Because it enables us to interpret how other organisms perceive the environment, and they don't have the same sensory systems we do. But until we're able to collect that data cheaply, we're limited in how much we can learn about the interactions between organisms. So these sensors are changing the way we are able to understand the environment, and therefore they change the way we understand how other organisms perceive the environment, and we can do something then to help or improve the situation.
Grazie.
Thank you. We now are going to talk about the future, in a way, in that it seems to me to understand that if we can synthetically replicate a number of mechanisms, potentially, in the future, it might be possible to create anything. You could make fuel at home, produce fuel at home, or medicals, which are fundamental, which are critical drugs for us. So is it really so, or am I exaggerating in trying to see what comes? Can this be a vision for your future research, or am I wrong?
I think that's a vision. That's a good vision for the future. So you can imagine that the technology, which we are far away from, I mean, we're not producing this now. So this is just fiction, but it is an interesting direction, because the idea would be, if you can create these kind of artificial systems based on what biology is doing, because biology can produce millions and millions of interesting molecules for us, but for their own purposes. So now we're going to create these artificial things that are producing the molecules that we want, whether it's fuel, whether it's pharmaceuticals. This is a potential future, really, for this kind of technology. And it's going to be, if this comes to be, it's going to be radical. We're currently living in difficult times that are changing very quickly. And, you know, there's an interesting book by Martin Gurri called The Revolt of the Public. I don't know if you know this, where he claims in the year 2001, humans have created as much information in that 1 year as in all of the years previous to that. So there's a huge revolution in information, which has become democratized and has created many of the problems that exist in the world today because of this. Imagine, as you're suggesting with these future visions, if technology for producing fuel, producing medicines that we need for a better life, has also become democratized. What does that change? How does that change the social structure of our world? And it's going to radically change the social structure of our world. So, on one hand, we are thinking about how to create the new technologies, and in parallel with that, we have to also consider what are going to be the potential social consequences of radically changing our socio-economic systems, which it certainly will.
Can I add to that?
Of course.
I hope for a future in which not just the economic and the social aspects of technology are considered, but the environmental ones. And a large part of our problem is because we manufacture too much and we dispose of too much unsustainably. And one of the great advantages of biology is that it's figured out, well, it doesn't figure it out, it is naturally sustainable. It reuses its own waste, it's very flexible and adaptable, and when something's finished with, it's not even a costly process to recycle. The other life lives on the remains of something that's passed away. And so my hope would be that if we're going to develop new technologies, they're not based on these old manufacturing paradigms. Yeah. Instead, we're finding ways to manufacture things we need for as long as we need them, that as soon as we don't need them, every single molecule goes back into the cycle that will enable us to use it again.
Ed è in parte collegato alla domanda.
So this is partially related to the next question. So we are changing about a change in the paradigm, and it is also what your research project is proposing. So tomorrow, if we have to face climate changes or problems related to lack of food, for example. So we have to change paradigm in the way in which we tackle these issues. Isn't it so?
I think so, and that concerns me a lot personally. So to take one example, I was saying I like to use technology in such a way that I can withdraw the technology. But it bothers me that I do something, or my lab does something like track a single insect's motion on a flower using a small computer about the size of a mobile phone that is essentially unrecyclable, and there are millions of those manufactured every year. I use that to collect data. That data I store on a supercomputer that has to be transmitted around the world. I then apply machine learning algorithms to trace, to find the location of that single insect in a photograph. And I've got thousands of those photographs. Every time I run that algorithm on a supercomputer in the US somewhere, I'm spitting CO2 out into the atmosphere, I'm generating heat, the machine needs to be cooled. So whilst I say, hey, you know, this really concerns me, we don't currently have another way. Yeah. And as a computer scientist, I could say, well, I'm not doing computer science anymore. But I can't improve the world if I don't do that either. But I realise that by doing it, I'm damaging the world. And we were talking before about this device that many of you have seen, a little scarecrow that you can buy at the hardware store or on Amazon. And it's like a little plastic owl with a sensor, and it's got red LED eyes, and it scares away birds, right? Now, that device is manufactured somewhere in Asia where people are struggling to get enough money to eat. It's made from non-recyclable materials. It consumes energy. It's not biodegradable at all. And it's designed to scare birds out of your back garden. Now, is that really a good use of technology? I don't think so. And so my hope is that we can move away from things like that. And in some cases, the old technology, a scarecrow made out of straw and bits of wood, we should now be able to recognise that that's a better approach. You know, so I think as a computer scientist, I try and find ways that change how we think about what we do. And sometimes I struggle with what I do. And sometimes I think, well, this has really helped me see things in a different way. And so I can share it with people. And hopefully you won't buy those scarecrows when you go home. You'll think, hey, what was that Alan guy from Australia with the weird accent saying about these things? And maybe I'll change people's opinion.
Stiamo ridendo perché ho un problema di piccioni sul mio balcone.
Well, we're laughing because I have a problem with pigeons on my terrace, and we were talking about that. I was going to buy that ultrasound scarecrow or owl, and I hadn't actually thought about all the issues that could have in terms of the surrounding environment. So thank you. Now, let's come to some conclusions so that you can have some questions later. So, let's go back to ACDC. Well, I've taken up a sort of a futuristic approach, but as we are getting to a first conclusion of this project, so at what point have you arrived, and what is the next step?
Those of you who are not in this profession, it's a European project, which means it ran because of COVID, it's run for 4.5 years. These are the last few days of the project that you're enjoying tonight. It's a collaborative project. Many different partners from different member states in the European Union have collaborated with us. And we are able to make some advances towards these artificial cells, but making specific molecules and specific functions. Again, far away from evolving cells, but some of the basic molecules, functions, and we're able to reproduce in the laboratory, so we made some progress. The next steps are already, let's say, proposed to the European Commission. So the project has now already put together plans. We've written out the plans for what's next, because we can produce these functional artificial things that are cell-sized, on demand, and they work. So one avenue that we proposed is, okay, we're going to make these artificial cells, like I just explained, that are going to be reporting on cancer states in human patients, because there are biomarkers that hospitals use, but there could be more sensitive techniques. So can we use these artificial cells that we already have produced and make them specifically sensitive to cancer markers, so they can be used to detect cancer at a very early stage? That's one project already written. Second project already written is, again, it's a bit related to human health. Can we use these artificial cells and embed them into a specific kind of organ and tissue that enhances the function of that kind of tissue? So in this case, we're working with a person who's working with brain tissues. So can we have brain tissues that we're examining in the lab together with our artificial cells that are helping to process signals, information transfer, things like this, or just the general health of the tissue? Can it promote the general health? So those are two next step projects that we already are hoping that will be funded in this area.
E arriviamo alla mia...
So my final question, and then we will have questions from the audience. And then we will have a surprise at the end, so don't go away. So the final question has already been mentioned by Alan, and has to do with the risks that every technological progress has had in the history of humankind. In this case, above all with reference to synthetic life system, we are speaking about a very hypothetical future, but what are the risks in both of your sectors? Because so you're working in the research field, but you also wonder where to get to and who should regulate, who should take decisions about how to regulate this very complex scenario made of different questions and different needs related also to the future of mankind.
Thanks for the question. There are some people here in the audience from my lab group, and they probably can easily tell you that even under ideal conditions, the system just breaks down and doesn't work. So the systems we're talking about are very primitive. They're not autonomous. They don't evolve. Right? They're very limited, and oftentimes they don't work, even under ideal conditions in a laboratory. So as far as the risks, right now the technology is so early and primitive, there aren't many risks. Now, considering the future of the technology, where you have a more of a distributed technology that people, non-experts, are using, this could be quite risky, and there have to be a lot of ethical considerations of what is the technology, what's the potential risk, and who controls this technology. These are very complicated problems which would require several more evenings of discussion to fully elaborate, but are essential for the next steps. But thankfully, I can say that this kind of artificial life that we're speaking about is very, it's very primitive. In fact, if you came to my lab and I show you, oh, look, we're doing this and this and this, you'd probably be like, oh, that's very boring. There's nothing to see here. But that's just the first steps, and that's fine. So, for example, if we are to, the first stage of AC/DC, everything we discussed tonight is fundamental research. If we're able to take that research and, for example, have better diagnostics for cancer, then that's an application. So that's the next step. So we're trying to understand, we built a car in our garage, and now we want to take the car out of the garage and see what happens, how it works. And targeting major health issues is one way we want to do it.
Sì, intendevo che il tema della biologia.
Well, I meant that the issue of synthetic biology is widespread throughout the world. So in the future, it will have to be tackled at a world level. This is what I meant.
Yeah. But that's going to be a much different version of this than what we're speaking about for this project. So I don't want to speculate too much. But then we have, but I think it's more concerning about not what is the potential use of the technology, but who is using the technology, who's controlling the technology. That's my basic answer. Yeah.
So it's a bit different in my area. The damage is largely already being realised, and when you look at the vast quantities of e-waste that we all generate, if we replace our mobile phone every 2 years, or we get a new TV because we can now get a bigger one, that technology ends up in developing nations being recycled by kids who are breathing toxic fumes and living under dreadful conditions. And I think smarter use and reuse of technology is my hope. How do we go about it? I think one important thing that I've learned from artificial life as a field is to value the perspectives of experts from many different domains. If you work in computer science, even in my faculty, which is quite diverse, it's very easy to fall into the trap of talking to other computer scientists about your work. But when you work across fields, so computer science, biology, ecology, physics, chemistry, you start to understand that as an individual with expertise, you can't solve any of the world's important problems. And so what I hope as a way forward is to collect people with diverse backgrounds, but with the common goal of sustaining the environment. And a good example of that, I think, is looking at how Europe is attempting to regulate the strategies for maintaining insects, which is an unusual thing, but it's been recognised relatively recently that many insects are in great peril. And whilst the data is a bit patchy, and some of the claims have been overstated, so you've probably heard about insect Armageddon, for instance, I don't think there's any doubt that either insects are doing just all right, or pest insects are going through the roof, and insects we need, the beneficial insects, are declining. And so what's happening is, I was recently at a conference where there are people like me, computer scientists, engineers, ecologists, community activists, policy makers, government representatives, all at this conference, all talking about what should we do. And I think that's the best thing any of us can do, is talk to people from other fields who are experts and work together to solve these big problems. And I think that applies, and I learned that from Artificial Life, which is a very diverse community of researchers.
Grazie, e direi che vi abbiamo
Thank you. I'm sure that we have raised interest, and people are certainly curious about a number of things now. So the floor is open to the audience.
This gentleman.
Può aspettare un secondo così...
Le devo dare il microfono perché sennò le traduttrici di là non riescono
Okay,
a
yes.
sentire, mi scusi. Prego.
Okay, how are these cell-like structures looking like? Most like eukaryotic cells, prokaryotic cells, differentiated cells, non-differentiated cells, or what?
Yep. So I was very vague, and we are open to any kind. So some of the artificial cells we make are microscopic, like a bacterium, and some are macroscopic, like larger than the largest eukaryote. So the size, because it's all artificial, we can make the size over vast scales. So that way, we can play with those parameters. But it's more like, to answer your question, it's more like eukaryotic cells, non-differentiated for the most part, which that means we have a cell membrane, and inside we have various specialized organelles. So it's more like a eukaryotic cell.
Okay, thank you.
You're welcome. More questions from the audience? Yes, this gentleman here.
Maybe it's a bit too specific because I don't really know what's going on in the lab, but for what I've gotten, we are able to create what we call a cell, a membrane with different specialized compartments. And those compartments, there are some that create energy, some that can use them. For example, I've gotten that these cells are able to move around in their environment.
Right.
Okay. What I wanted to ask is this movement that they are able to do is like a random movement, or can they do like something moving towards a specific signal, like chemotaxis?
Right. So thank you for the question. Both. So we have some systems that seem to be moving randomly, and they tend to be larger than what you would consider for Brownian motion. But anyway, so it's random walks, let's say. And then we have chemotactic ones as well. So they're sensitive to the environment. They can sense chemicals in the environment, and they can move directionally in these gradients. So it's chemotaxis. So what we're talking about here, if you're not specialized, you think about when you're hungry, you have all the senses to smell where the food is, and you can walk towards where the food is, and that's how you sustain yourself. In principle, that's what these artificial cells can do. They have sensors. They're very primitive, so they can't smell, steaks cooking or something like this, but they're very simple. But, and then they could move directionally towards the source of these chemicals. So we have both kinds of motion.
Yeah, and a follow-up to that, I wanted to ask, since we are not we don't really care about how similar these mechanisms are to what nature has come up with.
Yeah.
The one that we are actually able to replicate, which replicate is not really the word I'm seeking for, that the mechanisms that they use in lab, are they the same that can be found in nature, or are different? Because from what I gotten, we don't really care about how the function is done. We just care that they can actually do this. So,
yeah, that's right.
This is basically.
Okay, the mechanisms are completely different from what biology uses. That's the answer. So, because they're so simple, the things that we make are very simple. If we consider what a cell is doing, it makes proteins that can sense different kinds of things from the environment. So these proteins are on the outside of the cell, and they recognize this. And then that sends a signal into the cell, which makes different kinds of proteins, and they rearrange themselves so the cell can move towards some chemical or something like this. This is all programmed by evolution and things like this. Our system is not. It's very simple. So it still senses the environment. It makes an internal rearrangement that allows it to move in that direction. But it has nothing to do with proteins and things like that. So it's very, very much, very much artificial in that sense. But it does the same thing. It's a system that could sustain itself by following gradients of chemicals, like we do.
Yeah, thank you.
Yeah. More questions over here.
The ice was broken.
Yeah, it's good. I don't know if he can ask a question.
So my question is related to artificial intelligence, and I have a question that crosses both your field of study, because a few days ago I was reading the possibility to use biological neurons for a computational brain, but not made of electronic neurons as today we do, but made of real neurons created in laboratory. And the first question for you is, are we far away from replicating an entire brain in laboratory, so in a vitro brain? And for you the question is, can the possibility to have biological neurons instead of electronic neurons that are very more powerful could be a breakthrough in the approach of artificial intelligence? Because today we are limited on computational resources, and in your opinion, could be useful to use real neurons instead of electronics ones in order to overcome this problem?
Do you want to start?
Sure.
Yeah.
Good question. Thank you. I immediately saw one amazing benefit. Biological neurons are biodegradable. And whilst I'm not enough of an expert to say that they would outperform electronic computers at many of the tasks, I can see them being useful if they're well engineered for achieving tasks that organisms achieve. So when you think of all the things we can do that computers can't do, we tend to kind of reassess what it is that makes us unique and special in our eyes as computer technology, electronics, improves. But there are some very basic things that have eluded us until we started looking at biological brains. And so there are groups here who are looking at trying to understand how those processes operate on, at a biological scale. So unfortunately, I can't really answer your question about whether or not they would be superior in terms of, maybe Martin can, our computation. But I can say, that I would actively support, promote encourage people to be looking at alternative ways of building these artificial neurons that are biological in their materials. Martin.
I want to just tell you a story. Our friend and colleague Takashi Ikegami in Tokyo, who's also part of this artificial life community, did an experiment. He made a little robot with wheels and sensors, and he had an electronic layer, so everything is a robot so far. Then what he did was he grew neuronal cells from mammals, I don't remember if it was humans, I just say mammals, on top of the electronic layer, so that the neural processing, so the processing from the sensors to the wheels, has to go through the organic cell layer. So he did this experiment, and he had these robots moving around in these mazes and things like that, and they were able to it was working. Now the issue is that does it work better with biological cells? That is not clear, that particular system worked better with biological cells. But actually, one thing came out of it that was very unexpected. So imagine the robot is coming towards an obstacle, and there's a sensor, so it knows it's a proximity sensor. So it starts to activate electronically the neuronal cells. Say, look, we're getting close to something. And then the robot would then avoid Alan, right? But it was not programmed to do it. But it was moving around and it was avoiding things without being programmed to do it, because the explanation that Takashi has, who did the experiment, he said, well, the neuronal cells didn't want to be activated. They just wanted to be left alone. So the issue was they were responding like a brain, let's say, but in the sense that they didn't want to be excited. They wanted just to be quiet. And so when they got excited, they had to make a decision to get out of there before they get too excited. So that's the layer of biology that you will inherently get with that kind of question. You have to also consider, okay, we can use this as a technology. Is it better or worse? But what does biology bring with it? It's going to bring something more than what you expect into these kinds of systems. But yeah, it's an open question, but there's some small experiments where they are trying this.
Thank you.
Thank you for the question. Some questions up here?
Yeah, okay, it's me again. So you said that the mechanisms are quite different compared to the ones you can observe in nature.
Right.
So I was wondering, first of all, I think you are in a full control of the DNA you can put into this kind of cells. I imagine you put way smaller amount of DNA compared to an eukaryotic cell. I mean, there is no size for average eukaryotic cells because they can vary a lot.
That's true. Yes, true.
That's
yeah.
true.
Yeah.
I think but by the way, I think should be less. I was wondering whether, you said it's not based on proteins. Do you have any, first of all, transcriptomic study on this DNA, whether it is expressed or not, and maybe epigenetics or metabolomics, if it's somehow activated and produces the metabolites that are somehow related to the pathway you are putting into the cells, for example?
Yeah, that's a good question. So there are different flavors of these. Some of our artificial cells, for example, the ones that move chemotactically, they don't need any DNA or protein. They're just fluid dynamics, so it's more physics related. The ones that are producing proteins, we either supply the proteins already made, so there's no transcriptomics, or we do supply DNA, usually in the form of a plasmid that needs to be expressed, usually with T7 polymerase, blah, blah, blah. So you do some transcription. But it is from, like you said, a small amount of DNA. So you have the expression of 1 or 2 genes, right? That's it. That's your genome, if you want to call it a genome. So, so yes, we have some quantitative data on these super simplified systems and how they're producing, you know, the RNA part, how they're producing the protein part. And also, it's interesting that when you're producing, let's call them biological parts in an artificial setting, they basically don't function very well. Yeah, that's one thing that evolution gives us. A living cell is a complete system, produces things on demand, let's say, in a very efficient way. There are very few side products that are problematic. In In the artificial stuff, many of the things we produce are just not functional. So there's a small proportion that actually works, and that's what we use to have the function. So in that sense, you see how far away we are from producing something that's really like a living cell, because a lot of the stuff just doesn't have the right environment to produce the functional molecules that biology does.
So you can also provide ribosome, for example, and have the transcription and maybe translation and splicing also?
That works. Splicing we never tried, but yes, we can do the transcription translation. We can use either a completely pure translation system or just cell extract, which is from squished cells. So we have both things working in these artificial cells.
Thank you.
Thank you for the question. We have a question up here.
Yeah, so I have so many questions for Martin, and I'm so lucky that we work very close by, so I won't ask you the question because I have to come to your lab, but I will profit from Alan's presence because I have a question for you. So my question is, do you think that your work is in the direction of, so I've always had this question, is intelligent life and the technological civilization sustainable for a planet? So does it mean that if we are able to solve the problems we have on this planet, then probably life is sustainable? Because I'm not sure that a planet can sustain intelligent life for so long.
It's a good question. I'm fortunate enough to live in a country which has been continuously occupied by the indigenous people for 60,000 years. Maybe it's not indefinitely sustainable, but 60,000 years is a pretty good run.
Dinosaurs had it longer.
Yeah, but the dinosaurs didn't last so well, did they? Let's
Billions
not talk about that. That's a bad example. No counter-example. But I would argue that the dinosaurs probably didn't develop technology like the indigenous Australian population did. I think it's underappreciated how effectively they managed the landscape and to what extent they managed the landscape. I think their engineering skill and understanding, like conscious understanding of the way in which they impacted their environment is also underappreciated, at least by white Anglo-Western science, and that dominates in Australia as it does in Europe. So there's a living example, I think, and the way we've chosen, well, no, we haven't really chosen. The way we've gone, and whether or not it was a decision, I suppose, is a different thing. The way we've gone is not sustainable. I agree with you. But I think there's at least one example that is. I hope that answers your question somewhat. This is like at the end of a lecture, where, right, everyone understood everything and they don't have any questions.
When dealing with information, which is the bigger obstacle? Spreading of information inside an artificial cell or outside, like communicating with other systems, other artificial cells?
I don't think there's a limitation from practical experiments that have been done by us and by other groups, by the way, who are also doing this. I don't think there's a limitation of information distribution within the cell and then between cells. I think both have been demonstrated. So that's not a problem. What's your concern?
I was thinking about the project that you told before about using artificial cells in human tissues.
Yeah. Yep.
Like, what would result from an immunitory response? Like a graft of artificial cells could be seen as a foreign organism invading a body. So how do we deal with this issue?
Yeah, there are some technical solutions for how to deal with making the artificial cells, let's say, invisible to the immune response. If that's what you want, maybe that's not what you want. Maybe you want, actually, the artificial cells to be functional for a small period of time, and you want the immune system to remove them. That's the other way of thinking about it, right? Maybe so it depends on what your experiment is and what your application is. But there are technical ways to make these artificial cells basically invisible. They're called stealth, stealth technology. So that doesn't seem to be, that is a concern, but probably something technically that we can deal with. Yeah. But again, you might not want the organism to ignore your artificial cell. Maybe you want it to destroy the artificial cell. Maybe that's the technology that's more interesting, actually. Mm-hmm. It's like you take a medicine when you're sick, right? You don't want the medicine to stay in your body forever. You want it to stay in your body. Like my friend here has a cold. I gave him some drugs this morning, and that lasted 4 hours. Maybe that's all you need, is an artificial cell that lasts 4 hours. Yeah.
So I have a question for Alan. So you were talking before about the problematic of using unsustainable technology to try to solve some of the most pressing problems that we are facing today. So my question for you is, do you think this is something temporary, in the sense that are there already enough studies on, I don't know, new materials, new sustainable materials that we can hope that this is just a temporary situation that we will overcome with time, or are you more critical about this? Do you think it's something that we...
Some things I try hard not to think about because they frighten me. I suppose it's a bit like the former question that I was asked about the sustainability generally of technology. Even though I say, look, here's this example of people who have lived for tens of thousands of years in a way that didn't completely destroy the place. It changed the place, but it didn't destroy it in the way that we're doing. I can't see how we could get back to living like that. I don't think that's feasible. And I can't see an easy way to divert our planet from the way we currently manufacture and apply technology. And I can scream till I'm blue in the face, but it's not sufficient. And it certainly concerns me that we're rushing headlong into the impacts of climate change, and we have known for decades and decades that this was coming, and we've not been able to do anything about it. And I think COVID's a really good example of how suddenly things can come upon us and how little power we've actually got to mitigate their effects. And so we can sit here and we've got these amazing drugs now, and everything was kind of pushed through at a great rate for one single pandemic. And the tidal wave that's hitting us now it's going to dwarf that, in my opinion. And we've got whole nations being, drowned. We've got ecosystems on fire. Like Australia, the east coast burnt, just hundreds and hundreds of kilometres just burnt, and it's not able to come back because it burnt so hot. The fire is a natural part of our landscape in Australia, but not fire of such intensity. And so the bush can't regenerate in the way that it could have before. The coral reefs are being bleached and destroyed. Invasive animals are taking over. And, so technology, it's actually a little part of the problem. And yes, I think it's a concern. And because I'm a computer scientist, I can kind of focus on that. And the ecology that concerns me, insect pollination and insect-plant interactions, I can focus on that. But all that other stuff out there, like how do you change from using, copper and lithium in batteries and all that, I don't know. And it's outside my area of expertise. But it's certainly frightening. So I don't know is the answer. Yeah, but it scares me.
So thank you so much for your time and for this beautiful conference, which opened the mind of everybody in different ways, because we ranged from one sector to the other. and with notes of optimism, I hope. So we will meet in some time to understand at which point we are. Is that right, Lucilla? You will have to organize another evening. Grazie.
Thank you.
Grazie.